Okay, so the new features in the GGA and Genomatix software suite in July 2014. First of all, a short overview using a few slides, and then I will also show a few of the features live in the software. One thing that we added to the RNA sequencing expression analysis, which is available only on the GGA, is the differential expression analysis algorithm DESeq2, which came out some time ago. And so this was added here, so you do not only have Audiclavery and the old DESeq and the AHR as you used to have, but also the DESeq2. And the DESeq2 also comes with a few parameters that you can change here in the graphical user interface. By default, the statistical testing method is the Wald test that is used for DESeq2, but if you want to use the likelihood ratio test, which makes the behavior of DESeq2 more, well, similar to DESeq, you can also change that. The dispersion fitting method, which is used to, well, fit a curve into your expression data in order to estimate the dispersion for the transcripts, is by default a parametric method. This can also be a local method instead if the parametric fit does not work. It might not work, I haven't seen it yet, but you might get an error in specific data sets. Then you can use the local one. Or if you don't want to assume that there is actually a dependency of the dispersion values to the expression values, you can also just use the mean of the measured dispersion as the last option here. This also, well, leads me to two new analysis plots that you get in the RNA-seq expression analysis. One is the histogram of p-values, so you use a p-value threshold in order to determine whether a transcript is differentially expressed or not. And the histogram of p-values gives you an overview of the distribution of the frequency of the p-values from 0 to 1 with a bin size of 0.05. And if you see something like this, where you have an enrichment of the low p-value, then this is already an indication that you actually have a difference between your experiment and your control. The other new plot that is available is the dispersion plot. The dispersion is a measurement of the variability actually between or among the replicates in each group for the expression of a given transcript. And it is important for determining whether you have a differentially expressed transcript or not. So the higher your dispersion is, the higher the difference in the mean between the experiment and control group actually has to be in order to call this transcript a differentially expressed transcript. Now, what you see here is a plot where the dispersions for each transcript are actually plotted over the mean of the normalized counts, which is a measure for the expression of the transcript. And you see that there is some variability here. These are the black dots in the end. And the problematic point is if you have a small number of replicates, you might get a underestimation of the dispersion, like you see here, if you assume that the dispersion is actually dependent to a certain degree on the expression. And this underestimation would lead to false positives. Now, what is done is we fit a curve, and the shape of that curve is actually influenced by whether you have a parametric fit or a local fit or the mean fit. We fit the curve in here, which represents the assumption that we have actually this dependency of dispersion on the expression. And the raw data that we get here from the measurement for the transcripts are then well, changed a little, so they are actually shrunk into the direction of this fitted curve, and this is represented by the blue area that you see here. So they are not completely substituted by this curve, but they are well moved in order to get a better estimation of the actual dispersion for each of the transcripts. An exception are the dots that you see up here, which are kind of well, outliers with very high dispersions. They are not corrected into the direction of the fitted line here in order to avoid underestimating them again, and in this way also to avoid generating false positives. So that's approximately what this dispersion plot means. And this is similar dispersion plots are also shown for DESEC, for the old DESEC, and also for EdgeR. Okay, another new feature that we have is within the small variant detection program. You might be aware of the fact that we use SAM tools in the background here, and we have a new parameter for multi-sample SNP calling. Now, why would you want to use multi-sample calling, and what is it? Multi-sample calling means you call the SNPs not only for one BAM file, but for more than one BAM file, several BAM files in one go, and you generate one VCF file then for the SNPs and for the other small variants. And you might want to do that if you want to actually compare two or more files to one another, two or more samples, like for example in a cancer and a matched normal data set, in order to find well, variants that are maybe only present in one of the samples and not present in the other sample. For example, like if you're looking for somatic SNPs in a cancer data set. And you use that multi-sample calling because it gives you additional information as to the genotype at a SNP position where you find a SNP in the other samples where you maybe not find the SNP. If you called your samples separately, which you can still do, you get only information for SNP position, but no information about the genotype at that position in other samples where you don't call SNP. So you do not know whether you actually have a reference genotype in that other sample or just not enough information to call anything there. And with a multi-sample calling, you can actually differentiate between such a ref call and a no call. Okay, now the downside of that this multi-sample calling is that the sensitivity goes down in SAM tools, and by checking this option that we have down here, the sensitivity increases again. So in the end we have, or are supposed to have, no sensitivity cost if we do a multi-sample calling. So this is the, well, recommended way to actually do the calling if you have more than one sample. Okay, the next, also only available in the GGA, just as the small variant calling, is a change, a minor change only in the copy number variation analysis. Here we just have new versions of our analysis program. CNMOPS is now in version 1.10.0 and Control3C is in version 7.0. This does not affect the results very much in the end. Okay, now for those features that are available in the GGA and also in the software suite. One is a small addition in the transcriptome viewer. So the transcriptome viewer is a tool that can visualize the coverage expression values in exons and also in on splice junctions in a given locus after you mapped your reads to the transcriptome index on the Genomatix mining station. And when viewing such a locus, you could switch on and off specific transcripts also in the previous versions, and in the new version now you can switch them on and off actually by source. So we have 3 different transcript sources in our annotation: RefSec, Ensemble, and GenBank, and you have this option here in this control to select whether you want to see all sources or only one of the 3 sources that we have available here. Okay, the next is an addition in the Genome Inspector. Genome Inspector is our tool for actually generating correlation graphs for bed files which might contain regions, genomic regions, for example, regions representing peaks from a ChIP-seq experiment where a transcription factor binds. And these correlation graphs are generated in a way that you have one anchor set with, let's say, the peaks of one transcription factor and one or also several partner sets with the peak positions from other transcription factors, for example, or any other set of genomic regions. And you see such an example here. You might already be familiar with that. Here we have an example where we have the data from ENCODE where we have STAT1 transcription factor binding as the anchor and STAT2 and STAT3 transcription factor binding as the 2 partners. And we see a peak from the partner sets around here. Now what has changed is you see that only after you generated a list of correlations or genomic elements based on the distance of the correlation that you find here. So for example, if I wanted to extract a list of correlations with my STAT2 binding regions in a window of 200 base pairs upstream and downstream of the 0 point here, so like from here to here, I would get such a list, and from this list I can jump into the Genome browser to see what is actually there. And now here comes the new thing from this output where you can jump to the Genome browser. Now the input bed files that you used here for that correlation are automatically loaded as tracks in the graph. So for example, here we have the STAT1 and the STAT2 region. STAT3, there is no peak in that region. Okay, another addition is in the frameworker, so our program for finding common patterns of transcription factor binding sites in a set of input sequences, or for example in a set of promoters. And these patterns of transcription factor binding sites are called frameworks, also they're also sometimes called models. You can now jump from the model details view where you see the list of your models with the different binding sites that are in the model and the information about the distances among them. You can directly jump into the graphical view by clicking on the name of the model, and this model will then be shown in the graphical view in the, here in this example, in a set of promoters actually. The next addition is in the program common TFs, which shows you just the common transcription factor binding sites, again in a set of sequences, not patterns like for frameworker, just the common binding sites. We have an additional view, namely a common matches table, in addition to the graphical view and the match summary table that you might already know from previous versions. In the common matches table, you just have a listing of all your common matches that fulfill the quorum criterion and the other criteria that you set in the analysis, and you can export this list also in Excel format. In the literature and pathways section, there is a new task. This is called gene TF analysis. It is not completely new because you could well, use that kind of analysis already from within the Genomatix pathway system. But now you can also directly access it just inputting a list of gene identifiers. And what is important is you can input a set of genes that needs to include at least one transcription factor. So if you don't have any transcription factor in your gene list, this won't work. And what the gene TF analysis does for you, it generates a table using the transcription factors from your input list and looking for interaction information of these transcription factors with the, well, all the genes in your list. So in this case, you see the transcription factors are listed here, and all the genes, including the transcription factors, for example, ZFHX3 here, but also all the other genes here, are listed in rows, and you get information about interaction based on co-citation, just as you get in the Genomatix pathway system, and also based on additional information about binding site information. So if we don't have any co-citation information, you just get a gray box here. If we do have co-citation information and we have a validated binding site from an ENCODE ChIP-seq experiment, so that means the transcription factor actually binds in the promoter or maybe also enhancer of the corresponding gene that we have here, then we get the blue background, as you see here with IRF1 and IRF2. If we do have a co-citation and we have a predicted binding site, so by in silico prediction with our MAT inspector algorithm, you get a green box here. And if we only have a co-citation but no additional information concerning binding sites, then you get a yellow box. Okay, a few things about well, new features in the Genomatix pathway system. Two small things, you can now also input ensemble gene IDs as gene ID input in addition to the entry gene IDs. And you can also use microRNA IDs, as you see here, as input for gene symbols or names. A few more things within the Genomatix pathway system. You can now not only change the color settings in the pathway system. Some of you might prefer, for example, to have green as coding for downregulated genes and not blue as it is the standard, and maybe white as for non-regulated genes instead of a well, yellowish orange as is the standard. And in previous versions you could change that certainly, but this information was gone after you logged out. And now you can actually save your user default and reload it in a later session, so you can directly access that. In future versions, this reloading will not be necessary anymore, but you will automatically get your user defaults as soon as you load a Genomatix pathway system network. Another addition is an option for extending your gene networks with microRNAs. You already had some other options for extending your networks with other co-cited genes, or specifically with co-cited transcription factors, or with potential downstream targets of transcription factor genes, but microRNAs were not in the list yet. Now with a new update, you can also add microRNAs. This the button for actually adding them, extending them is in the navigation bar that you have at the bottom of the Genomatix pathway system interface, and just as you do with the other extension buttons that you have, you select the genes that you want to extend, and then you click on the button and you get your added microRNAs, and you can just click or double click on the nodes and also on the connections in order to see additional information, just as you can with the other gene nodes. One more thing in the Genomatix pathway system is the treatment of connections that are based only on validated regulatory evidence. Validated regulatory evidence being transcription factor binding in the regulatory regions of a gene based on ENCODE data. In previous versions, you could have connections that are based only on these ChIP-seq data without any other evidence coming from the literature. And in the new version, this option is deactivated by default. You can still activate it if you like to, and you find this in the settings actually. So by default you have this option activated, exclude validated regulatory evidences, and you have an example here in this network where we have a shortest path network that has a stat here at the center, and we try, or the program actually tries to generate a network starting from stat here and connecting all the other elements here in the network with as few hops as possible. So if possible, everything is connected directly to stat1. And we have three nodes here that are not connected directly, and for these two nodes the reason is because we do not show any connections based that are based only on validated regulatory evidences, and if we change that and recalculate the network, then we get the direct connections here as well, which are shown here to have well, in the end, no co-citations but validated interactions. Another new feature in the Genomatix pathway system is that you can now include gene families in the network. In previous versions, gene families were not recognized and were in this case also not part of gene networks. Now you can include or they are automatically included in networks. Here in this network you see, for example, metallothionein, which is a gene family, and reason for that is in many cases the actual term that appears in the abstracts of PubMed articles is not the specific term for a specific member of the gene family, so not for a specific metallothionein, and this would be lost if we couldn't resolve that in some way, and now we can do that. So you can see gene families included here. If you double click on a connection, you get the corresponding citations, co-citations. You still have the specific information about specific members of the families separately. Here, for example, MT2A is metallothionein 2A, and this has its own node and its own connection for those cases where MT2A is specifically cited in the abstract. Okay, so much for the Genomatix pathway system. Now three more small additions here. One is in the BAM file toolbox. You can now generate BAM file statistics for uploaded BAM files, or you can also view them if you generate the BAM files by mapping in the Genomatix mining station. If you have a GGA, the generated statistics will be automatically transferred here as well. These statistics include the pileup size distribution, include the distribution of the mapping qualities, and for paired end data it also includes an insert size distribution and the distribution of the strand orientation. In another toolbox, in the gene ID toolbox, you can now also use the ensemble gene IDs just as you can for the Genomatix pathway system as input, and you can also certainly mix different types, so mix entree and ensemble gene IDs. And lastly, in the list comparison, we now changed how we treat key value pairs where we have the same key more than one time with different values. Now in this case both values are reported in the output. If you look at this example where you have 2 times the key B and with 2 different values, 2 and 3 in the first list here, and you look at the output of the union of the 2 lists that you have here, you get both values actually associated with the key B. Okay, a few things also live in the program. Just to show you the effects, I have a transcriptome viewer data set already loaded here from the Genomatix mining station. This is an expression data set from TGF-beta1 treated kidney cells. And we are looking here at the actin beta locus with this splice graph view that is the standard view in the transcriptome viewer with the expression of the exons shown in color code and the expression of the splice sites shown in the thickness of the connections here. And now we have all of our annotated transcripts here in this view, and we have the control down here where we can now change from which source we actually want to see the transcripts. For example, in RefSec, we have only actually 1 annotated transcript in this locus, or here in GenBank we have a few more, and so this is just the corresponding subset. It's one thing. And yeah, next is the example with the genome inspector, where we have a result with where we have STAT1, transcription factor binding, supertrack, that is our anchor set, and this compared to STAT2 and STAT3 tracks also from ENCODE. And now if we view the correlations as a list, well, maybe you can take the STAT3 peak here, so this would be this curve actually. This goes from -300 to 300 under this peak approximately. So we enter the distance range for the correlation here. We get the correlations within that window. Takes a little until it is calculated completely. Not too long, I hope. Yeah, here it comes. Now we have that list of the correlations with well, the position, but no information about actually what is annotated there. And now we click on the Genome browser link. We jump to the Genome browser and can now see the annotation in that region. including, after it is loaded, including the peak regions for our ChIP-seq data. Here in this case from STAT1 and also STAT3. STAT2 is also loaded as it was part of the original correlation analysis here, but doesn't have any peak in this area. Okay, now to show you the gene TF analysis in the literature and pathways, I'll just open this here. You find it in this menu item here, the last item shown with a new tag. Okay, now what can you do? You can upload or enter a gene list. Either you enter it here or you upload it here. I will actually upload a file that we find here with just a number of gene identifiers or entry gene identifiers, and some of them are transcription factors, and that's why it actually will work. So I just upload this file with my gene identifiers, and I can specify the minimum evidence level that I want to see in order to get the colored background. The default is sentence level, but you can also be more or less stringent. Sentence level means that the gene and the transcription factor have to be co-cited in the same sentence in an abstract in PubMed. You can also have only abstract levels, so they don't need to be in the same sentence anymore. You can be more specific. Function world level means you in the sentence there also needs to be a function word such as regulates or inhibits or activates. Or you can go to the validated regulatory level, meaning that you need to have a ChIP-seq based information about the correlation, or to the expert level where you have hand annotated information about the correlation between, or functional correlation between the two genes. And you can also set the minimum evidence number, so how many co-citations do I have to have, for example. We just stick with the defaults here and submit the query, and this is very quick. So now we have the list of our correlations with our transcription factors here. You see that also cofactors are included, and with all of our genes, whether they be the transcription factors or not, here in this column, and we see now the information that is available. The legend is shown here, and so we have, for example, a lot of validated binding sites here for STAT1 with interaction with the genes here. We have in green without any validated binding interaction for IRF for some of them, and for BCL3, which is a cofactor, we don't have any neither validated interaction nor predicted binding sites. Only co-citation shown here in yellow with a number of genes. Okay, so much for the gene TF analysis. Then we have a few things in the Genomatix pathway system. I won't show the ensemble IDs and the Mirena ID input, but I'll show the user-defined color settings. So here is a network already preloaded with the standard color settings for red upregulated, blue downregulated, and you change the colors here. So for example, we use green for downregulated, and we get rid of the yellow here. And we can save that as our user default. And if we cancel that, nothing happens. Yeah. We can directly activate it certainly by just saving the settings, but we can also just load the user default. So imagine we just loaded a new network here, and you load the user default, and you will automatically get the new color scheme that you selected. Okay, just get back to the defaults here. So with the default settings, you can always switch back to the defaults. Now for the extension with microRNAs, you see the extension buttons down here. So for example, you have extension with just genes or terms that have frequent co-citations. We have transcription factors, etc. And here the third one now, shown with these well, small microRNA symbols in orange here at the bottom, now is for extending by microRNAs. And you select your genes. Oh, sorry, you can select them with a control click here. You do multiple selections, and you just click here. You get the usual parameter choice for number of elements that you want to add, and also for the connectivity. So how many co-citations do we have to have, at least for a new microRNA to actually to be added here. If we leave that at the defaults and just extend it, takes a few seconds until the microRNAs are loaded and added to the network. Now we got them here, and you can do a double click on any of them, showing you information about the microRNA, and also a double click on the connection will show you the co-citations. Okay, now for the well, validated regulatory evidence that is by default excluded, I'll show you another network. Here we have this network with our stat factor at the center, and we have the two elements up here which are not directly connected to stat. You see that some of the lines here are dashed lines. They represent those cases where we do have a validated interaction information, but by default they also need to have co-citations, which you also see in the tooltip text. We have 8 co-citations on function word level and 8 co-citations on sentence level, and this is also the case here. So we always have co-citations included. These connections here are also based on co-citations, but nothing directly here connected to stat1. And you change the setting for adding also connections that are now based only on ENCODE ChIP-seq data by going to this settings dialog using the settings button down here, and actually removing the check mark here in this box. Then you save the settings, and you need to reload the network by clicking the network generation button that you have down here. This will calculate the network again. And now, for example, for the PIBF1, which we saw indirectly connected to stat1 here, now we see that this is based only on validated interactions. In addition, we have a predicted binding site, but predicted binding sites never are enough to constitute a connection here, but we don't have any co-citations from the literature. This is useful if you have a network that includes maybe several transcription factors that are highly connected to other genes in your network just based on ENCODE chipsec data, but do not have a lot of co-citations. In this case, you will get a denser network, so to speak. On the other hand, if you want to avoid this network being dominated only by these chipsec connections, you use the default option in order to be able to see only those connections that are based on the literature. Okay, so the last point here in the pathway system is the example for the gene families. Here I actually uploaded only one gene, the one that you find here in the center, MTF1, and let the system automatically generate a network with the co-cited genes. And this included now also a node here for metallothionein, and it also includes here the node for MT2A, which is metallothionein 2A. And if you want to see the co-citations of MTF1 to metallothionein, here you just double-click on the connection, and you see, for example, here in the first citation, we have metallothionein without any specification which metallothionein it actually is, co-cited with our MTF1 factor down here. In contrast, if we look at the connection between MT2A and MTF1, we now see also, well, this is MT2 is an alias for MT2A, obviously, which is co-cited with MTF1, or here we have MT2A also co-cited with MTF1. So we still see the specific co-citations to members of this gene family as well. Okay, one more thing that I'd like to show you is how the statistics for your BAM files actually look in the BAM file toolbox. So the BAM file toolbox is to be found here under Tools. And if we open that, we can just select a BAM file and let the system generate the statistics for us. I already did that for these two BAM files, so you can view the statistics from your project management, where you have your BAM files also listed, the uploaded BAM files. And if we open this directory structure here and click on the link that leads us to the BAM files, we will see the statistics. Here is actually the start of the BAM file that you can view. You can download the BAM file here. And then you have the pileup distribution, where you have the number of pileups that you get for the different pileup sizes in the data. You have the mapping quality distribution here with a mapping quality of 60 as the highest value, and also some with lower values. You have, as this is paired end data, you have the insert size distribution here with a maximum at around 220 or so. And you have the strand orientation information, where you have plus minus orientation for the forward and reverse read in the mapped data dominating the statistic, and a few read pairs obviously with other strand orientation, which can be an indication of genomic rearrangements. You can print all these charts, and you can also download the charts in different formats that you see here. Okay, the last example, again, showing you the list comparison, how this is done. The list comparison tool is also found here under Tools. And for those of you who haven't used it yet, you can compare up to 10 lists in here. Depending on what you select here, you will get more input elements, and you can enter your list elements here, or you can also upload a file with your list elements. Optionally, you can also use associated values. So for example, gene IDs here and expression values here, or whatever you like. And I just show you this with this example where we have a list of 4 with A, B, B, and C in the first list, and with A, B, C, and D in the second list, and with, well, slightly different values here, 4, 3, 2, 1 instead of 1, 2, 3, 4. Now, if we do a comparison of the lists, and we have this situation where we have the same identifier more than one time with different values, we get in the output, apart from the Venn diagram that you see here, we get, well, the options for downloading different, different combinations of the list, the union, or what is the intersection, or also what is just in list 1 or just in list 2. For the union, for example, if you download that as an Excel file, you now see that from list 1 we have both 2 and 3 associated with the element B here, and they are both shown. Okay, so much for the overview of the July update of the GGA and the Genomatix software suite. Now we still have, well, a few minutes, maybe 10 minutes, so if you have questions at the moment, I'd ask you to ask them. I have unmuted you, so if there are any questions, please go ahead. Okay, I hear no questions, so I'd like to thank you for your attention. I hope it helped you a little bit and maybe gave you some ideas about what you can do with the new version. And well, I wish you a nice day, a nice evening, and maybe we will meet again or hear from one another again in one of the next web seminars. Bye-bye.
Bye.